Yes, I think that the biggest improvement in survival in cancer through new treatments is melanoma. Melanoma is the one disease that we have had a huge breakthrough in survival. And if I would see a patient 15 years ago, they'd come to my clinic, they'd have stage 4 melanoma. I would basically be telling them, we don't have any therapy that we have a statistically significant improvement in survival that we can give you. But we can give you treatment to help your symptoms, make you more comfortable. But for stage 4 melanoma, the average survival was from 10 to 12 months. So it was a very dismal scenario. But there were glimmers of possibility for advance based on Steve Rosenberg's work at the National Cancer Institute. He was chief of the surgery branch during the time I was there doing my fellowship. And in fact, he's only recently retired, but he's still very active and involved in immunotherapy for melanoma. And he had pioneered the use of interleukin 2, which is a signaling molecule that our own body makes. The helper T cells, when confronted with foreign invaders, works with the macrophage to set up a signaling circuit to the killer T cells. And one of the key signaling molecules is interleukin 2. And that can activate the killer T cell to kill the cancer cell. So you could have T cells on top of the cancer cell, and they want to kill that cell, but they need the interleukin 2 signal. It's a required signal to get them to release their bombs, gramazine B and porphyrin, that can kill the cancer cell. And so he started this investigation into interleukin 2 in the 80s. And it was shown that it could be effective, but only 10% of patients had durable remission. So 10% could be potentially cured, but that's too small a number to achieve clinically significant changes in overall survival. And then trials looked at adding chemotherapy to the interleukin 2. And I did that for many years. It was called biochemotherapy. And it did have a 50% response rate. So 50% of the patient's tumors would shrink by at least 30%. But in the end, this overall survival was not statistically better. And it was a very tough treatment to go through. You had to be an inpatient, 4 days in the hospital, feeling like just terrible for another week, and then back in 3 weeks to get the therapy again. So it kind of dominated their lives. It wasn't like, their lives were revolving around this treatment that wasn't statistically significantly better, but did have improvements in quality of life in terms of tumor shrinkage, decreased symptoms. So no chemotherapy has ever been found to change the natural history of melanoma. And this is thought to be because melanoma cells are like stem cells. A stem cell is a self-regenerating cell. And in our bone marrow, we have stem cells that make our white blood cells and red blood cells and all our different circulating blood cells. And when we give chemotherapy, the stem cell pool survives the chemotherapy. The cells that are no longer stem cells and are circulating can be killed by the chemotherapy, so the white blood cell count goes down. But within 3 weeks, the stem cells have repopulated the white blood cells, and the person is safe from infection. And that's one of the reasons we give chemotherapy every 3 weeks in general, because that's how long it takes the stem cells to recover, to refurbish our cells that are circulating in the blood to keep us safe from infection. So you can imagine if the cancer cells have the same qualities as the bone marrow stem cells, and they are resistant to the chemotherapy, both cell types become resistant. So cancer cannot be cured if it's a stem cell type cancer with chemotherapy, because you'd have to give so much, you would be killing the stem cells in the bone marrow, which you need to recover your normal cells. So it was important to find a new way forward, and it was the breakthrough by Hanjo in Japan, who discovered the sword that sticks out of the cancer cell and kills the T cell that's on its surface trying to kill it. And so that sword was a Nobel Prize winning discovery in his lab in Japan. And then the nivolumab was developed as a drug, and Keytruda was developed as a drug. When the sword is coming in to attach to the receptor, the antibody stops the sword from killing the T cell. So it's like a shield protecting the T cell, and now the T cell can release its bombs and kill the cancer cell. So that was the huge breakthrough. And in addition, as we talk about interleukin 2, the first immunotherapy drug to be approved was the CTLA4 antibody. And that's over with the macrophage and the helper cells working together to send the interleukin 2 signal to the killer cell. And when they're working together, there's an off switch that turns on, and now they aren't engaged, and the interleukin 2 goes away. So now you aren't sending that signal to the killer cell. So the CTLA4 antibody stops that off switch. Now the two cells work together and are sending interleukin 2 all the time to activate the killer T cell. And so that was the first treatment to improve survival ever for melanoma. CTLA4 antibody, Yervoy, or ipilimumab. And then Bristol Myers Squibb developed nivolumab, which was over here with protecting the killer T cell from the sword, in addition to giving the Yervoy, which keeps the interleukin 2 going. And that combination of immunotherapy led to this dramatic improvement in survival. And the 10-year overall survival data were just published in the New England Journal of Medicine on January 2nd, this month. And what that showed was that the melanoma-specific survival at 10 years was 52%. And, so that means that we've gone from a survival of 10 months to 50% of people being alive at 10 years. So this is the biggest tectonic plate shifting result for developing therapy for melanoma. Now we understand that the individual patient's tumor and the individual patient's biology, why are some people cured and other people aren't? So now there's a lot of work being done to determine the molecular elements of the immune system interaction with the tumor. And we can predict, based on certain biomarkers and blood tests, who's most likely to benefit from the immunotherapy. And the other class of therapy that was developed was BRAF targeting therapy. And vemurafenib was developed by Genentech, and it emerged just after Yervoy. Now here's a targeting drug that binds to the BRAF, which is a circuit in the cell. So there's receptors that get a signal. The signal goes into the cell, and there's different pieces that are activated in a circuit. And BRAF is one of the second pieces of a circuit in response to these signals. And the signal goes away, the BRAF turns off. But in about 50% of melanoma patients, this BRAF is mutated and it's always on. Doesn't matter what's going on up above. It's just sending a signal to grow. And so that's a carcinogenic sort of process, oncogenic, in melanoma that has this BRAF mutation. And it turned out that most melanomas that have that mutation are addicted to that signal. They need that signal to survive. And so when vemurafenib was given, 50-60% of the patients responded to the treatment. And survival was improved from the 10 to 12 months that we talked about out to about 14 months. But it was only working for a few, a while, and then it would wear off. Why does it wear off? And so there's the BRAF is signaling to this MEK, and the MEK is signaling downstream. And it turned out that in addition to BRAF, there's CRAF. And CRAF could go around the BRAF inhibition and turn on MEK, and MEK would keep the cell alive. So they added to the BRAF inhibitor, they added a MEK inhibitor, and that was first developed by GlaxoSmithKline. Dabrafenib is the BRAF inhibitor, and trametinib was the MEK inhibitor. And now people were living much longer. I've had some of my patients were cured taking these pills, although the cure rate is lower than with immunotherapy. So immunotherapy is the primary treatment strategy. But if you do fail that, you can get BRAF and MEK treatment if you have a BRAF mutation. So you can have a sequence of therapies. And then there's another element to the treatments for melanoma is anti-angiogenesis therapy, which kills the blood vessels that feed the tumor. So the tumors require a blood supply to continue to get bigger and bigger. And there's one drug called Lenvima, which we use in kidney cancer, and it was found to have benefit also in melanoma. And it could be given in combination with Keytruda, the PD-1 inhibitor that's working through the immune system. And now we're seeing that in kidney cancer. Lenvima, Keytruda is a good combination, as it is in melanoma. So I think one of the great things is we have this breakthrough with the combination of nivolumab and Yervoy. And then we have newer ones, they're LAG inhibitors, and Opdualag is a combination of nivolumab and a LAG antibody. And then the recent study that was presented at ASCO was combining Yervoy, the LAG antibody, and the PD-L1 antibody, nivolumab. And those three together were found to be safe and effective with a long period of survival at 2 years of about 70%. So, and then they've combined Yervoy, low-dose Yervoy with Keytruda, and that also has about a 75% 2-year survival. So one of the keys is, all right, we have these tools in the box. How do we know which person gets which tool? What's the best tool for the given individual? And that's where the biomarker testing comes in. And for all of my patients are blood biopsies, gene sequencing, and we can measure how many swords are there present on the cancer. So with immunochemistry, we can look at a tissue section from the melanoma and see, oh, this has a lot of swords on it. That patient's very likely to respond to an immunotherapy drug. And when you give 2 immunotherapy drugs at the same time, there's much more toxicity. So if we can analyze the tumor and the gene sequencing of the circulating tumor DNA, we can refine our treatment choices. High PD-L1 or lots of mutations in the tumor, we don't need to use both immunotherapy drugs or 3 immunotherapy drugs. Just one is enough. And you get the same benefit with much less toxicity. So now we're sort of on how do we refine the application of these drugs by directing the right treatment strategy to the right person's genetic the tumor makeup. The genetics of the tumor are helping us to figure out how to approach that individual patient.